Hello everyone, I hope I'm audible at the back. It's my pleasure to welcome and introduce Professor Ravi Mahalingam, Department of Neurology, University of Colorado School of Medicine, United States. Dr. Ravi Mahalingam will deliver a talk titled Varicella Zoster virus is the risk factor for Alzheimer's disease. Let me read out a short bio of Professor Mahalingam. Dr. Mahalingam is a professor of neurology at the University of Colorado School of Medicine. He is also a visiting professor at Sri Ramachandra Medical College and Research Institute, Chennai, India. He did his master's in chemistry at the Indian Institute of Technology in Mumbai, India, in 1976, and completed his PhD in biochemistry and molecular biology in 1980 at the Southern Illinois University, California, Illinois, United States. For the past 3 decades, Dr. Mahalingam has been working on the pathogenesis, latency, and the reactivation of human varicella zoster virus. He established a unique animal model using Simian varicella virus infection in non-human primates. In collaboration with Professor Wayne Gray, Dr. Mahalingam sequenced the complete primate virus genome in 2001. His laboratory currently focuses on how herpes virus infections increase Alzheimer's disease risk in the elderly. Let's welcome Professor Mahalingam again.
Thank you.
Dr. Mahalingam.
Thank you. Thank you for the very kind introduction. I see a lot of students here. So are there anybody who's familiar with virology here? No, so I can say anything I want. You can believe me. Because if someone was going to say yes, I was going to take them outside and pay them some money so that they don't have to disagree with me. So what I want to say is, anybody, you have any question or something is not clear, just stop me like a class and ask me, because I want to take you through this journey. And let's start with this. So this, I'm a faculty in a neurology department. This is our campus, University of Anschutz Medical Campus. And this is the outpatient thing. This is the inpatient. And my office is somewhere in this building right here. This is famous building called Building 500, where President Eisenhower was treated here. And the other side is the Children's Hospital here. Anyway, so what am I going to tell you today? This is what is the outline of what I'm going to tell you. First, I'm going to give you some background on herpes viruses. Then we will talk about VZV. I'll tell you what VZV is in a minute. It's called Varicella Zoster virus. And Alzheimer's disease. Most of you have heard about Alzheimer's disease, or at least you know some relative of yours suffering from Alzheimer's disease or dementia. Then we'll talk about animal model of VZV infection, which is the monkey chickenpox or simian varicella virus in primates. Then we talk about induction of amylin and amyloidogenic environment. The names may be a little, uncomfortable because it's a jargon, but let's go through this. And then how virus induces amyloid in multiple organs during virus infection. Then we will talk about the olfactory system, the sense of smell, and how Alzheimer's is related to that and how virus infection alters that. And the second part, very short, I want to present something, a novel which was published in the last couple of weeks, which was very striking. So I just want to sort of go through that quickly. So, herpes virus. Herpes virus, we all, would have heard that as soon as somebody says herpes, it is like there is a connotation, it's a sexually transmitted, or it is an elderly person having herpes. There are actually 8 herpes viruses, okay? Herpes simplex virus 1, 2, VZV or varicella zoster virus, or it's called herpes virus 3. Then there is CMV, herpes virus, it actually should be 4. And 4 is here, 5 and 6. These are categorized to alpha, beta, and gamma, depending on the structure of their DNA. Each group has a different structure of DNA. That's how they are classified. Just to quickly tell you what they do, these 3 viruses become dormant you get chickenpox or you get canker sores or genital sores, it goes to latency in the ganglia, or these are tiny, tiny organs along the spine. They are located there. And these cause genital lesions or recurrent meningitis, chickenpox, everybody knows, and shingles. These viruses, cytomegalovirus, human herpes 6A, B, and 6, they are called enlarged infected cells. That's what you see when they infect, or cytomegalic, that's the scientific term. They become latent in T cells. T cells are immune cells. And they also in secretory glands, kidneys, or other tissues, and they cause fetal growth defect, and they make what's called cytomegalic inclusion disease, or another disease called roseola. Finally, in the third group, Epstein-Barr virus, a lot of you might have heard about it because this has been linked to multiple sclerosis recently. And human herpes 8, which is the last member of this group, which is all Kaposi sarcoma virus, or it becomes latent in B cells or another kind of immune cells, and it causes Kaposi sarcoma or lymphoma in HIV patients. Okay, so this is a family of herpes viruses. Today, we are going to focus mainly on this one, which causes chickenpox in children and becomes dormant. So, as you can see, initially it causes chickenpox, and the virus travels along the nerve endings, this is the spinal column, goes to this organ called ganglia. And then when the person is more than 60, 70, 80, it can also happen in younger generation, but the virus leaves latency, it comes back and causes shingles or zoster. Now, this is the general pattern. It just becomes latent and gets back to cause shingles. This can happen along the neuraxis. In other words, from all the way to the head to the tail, there are ganglia. It can reactivate from any one of them and cause rash. As you can see in this picture, if it reactivates from trigeminal ganglia, it causes blisters in the forehead. If it causes in the cervical or thoracic ganglia, it causes blisters more in the neck. Then this is the lumbar ganglia reactivation. This is sacral ganglia. In other words, it can be in any location from head to tail, and it always moves from one area. As you can see here, it is completely localized here. This person would not have anything in the bottom here. It has only forehead. This person has only here. So we call this is belt of roses from hell, because there's so much pain, so much discomfort. But there is antiviral. We treat them and they feel better. Now, however, if the virus, instead of going back to cause shingles, if it goes back into the brain, CNS, it goes to the brain, or even brainstem, that is where it's bad news. It causes many diseases. See, virus latency, it reactivates, and zoster happens. All of this can happen without rash. Somebody will have pain, but absolutely no blisters. That is called zoster sine herpes. So when a clinician sees the patient, they have to be able to diagnose that as it is indeed zoster without rash. But when it goes to the central nervous system instead of the skin, it can cause many diseases. Vasculopathy is stroke, central nervous or peripheral nervous disease, GI, gastrointestinal disease, cardiac, pulmonary disease, and a lot of eye issues. People can go blind. So all of these complications can happen in the absence of rash. So this is a big challenge to the clinicians. But to get to the brain, does it not have to cross a barrier? Oh, T cells are the one that carries. The T cells always go everywhere. So the T cell, it's a T cell tropic virus. It carries the virus to think. And then in a lot of situations, there is a breakage of blood-brain barrier in these diseases, and that makes it even worse. So now I'm going to go to the part one of my talk, the herpes viruses and dementia risk. The relative risk of dementia, particularly with people who have ophthalmic or eye zoster, is 2.97. That's the extent of risk within 5 years after somebody had got zoster. And with any other part of the body, within 6 years, there is an 1.1. When you treat them with antiviral, there is an antiviral, it's called acyclovir. It dramatically reduces. And it also is the case that herpes simplex virus also developed dementia within first 10 years. The relative risk is 2.5, and when you treat them, it really comes down. So this indicates that there is definitely a virus contribution in terms of the dementia risk. Now, this was a very interesting study. They studied over a period of 12 years the cumulative incidence of dementia with both HSV and VZV, both viruses, is much higher when there is no treatment, and it decreases with treatment, and the controls fall in between. But definitely, this study proved that over a period of 12 years, usually the zoster happens usually from 60 years on. The higher the age, people who are in their 90s, there is more risk of zoster. 1 and 2, every one of us, if we get to be 90, 1 and 2 of us will get shingles. So, in general, if you compare Alzheimer's disease and VZV reactivation, these are all the factors in Alzheimer's disease. They share both these features, importantly, cerebrovascular disease and amyloid deposition, which is a key factor in Alzheimer's disease. VZV infection shares these pathologies. Okay? It turns out in the last just a couple of years, there have been more number of papers. I just selected only a few, and this was the very latest. This came out after I arrived in India. The recombinant shingles vaccine, there is a vaccine, they said that it associated with the lowering the risk of dementia. And we had a meeting in Alzheimer's Association about a month ago, and there were a lot of people from GlaxoSmithKline. They said, definitely with the vaccination, you can reduce the dementia by about 20%. This is just a preliminary result. In our own clinic, there are 2 cases. There was one case of vasculopathy or stroke, where this person has progressive multi-infarct dementia, and we treated the patient with acyclovir, the antiviral. That person definitely regained the mild cognitive impairment. And there's another very interesting story. This patient was a herpes virologist. He was one of our very highly respected herpes virologists. He used to come to the meetings, and suddenly his cognition was impaired. He was not coordinated. We didn't know why. Thankfully, my mentor, who is no more now, he's a clinician. He talked to him and he said, there is some problem. They said, oh, no, they said there is a cancer tumor. They are doing biopsy and everything. So my mentor said, wait, let me get the tissue. So we were given the tissue. We analyzed. It was the virus. So we brought him to the clinic. We gave him IV acyclovir, and he got better completely. So there are 2 cases in our own clinic. We were able to help dementia patients with acyclovir. Now, what is Alzheimer's disease? Alzheimer's disease is just a group. Okay. Now, there are other diseases like Parkinson's or vascular dementia. There are many dementias where some molecule, some protein misfolds and aggregates, become sticky, and starts becoming toxic and inflammatory. These are all the various proteins. But I'm not going to talk about all the proteins. I'm going to be talking about A, beta 42, and amylin. These are the only 2 that are really linked with Alzheimer's. So I'm going to be focusing on these proteins. These other proteins also form amyloid, but they go to other organs and cause other diseases. So amylin is made in the pancreas along with insulin, and it tells you, it goes to the brain and says, yes, you have eaten. It's a satiety gene. Amylin is made along with insulin in the pancreas, goes to the brain, says, yes, you have eaten, you're full. That is the normal function of amylin. However, amylin changes its structure in the brain and it becomes amyloid. So that's when you have problems even with diabetic people. And elevated amylin levels in the brains of patients with Alzheimer's disease have been noted. So let's start with how we make the linkage. This is virus-infected cell, neuronal cell, and we stain for viral protein to make sure that there is virus protein in these cells. Then we also stain for amylin, we stain for amyloid beta and Thio-T. It's a chemical. This chemical binds to anything that has beta-related structure. Amyloid has beta-related structure. So this is also amyloid positive, amylin positive, amyloid positive, A beta positive. And uninfected cells here does not have any of this. Therefore, virus-infected cells in culture produces amyloid. And another very interesting thing, all this work is done by Andrew Bubak. Dr. Andrew Bubak is a faculty in our department. Is an amazing.
Professor,
yes.
I have a question regarding the previous slide. What organism is this and which cells are in?
This is a neuronal cell in culture, human neurons in culture. Yes. So what Andrew did, a lot of the work I'm going to present is done by Andrew Bubak. I wish he was here one of these days. I'm sure he'll be here. Very bright young man. He showed that during acute zoster, when somebody has shingles, the plasma, the blood plasma, he found there is elevated levels of amyloid with A beta and amylin levels, and also it is amyloidogenic. What do I mean by amyloidogenic? If you take the serum, amyloid is nothing but A beta is all small protein fragments, right? It can misfold and become amyloid, right? So if you take the serum from these patients and spike them with A beta, immediately misfolds and becomes amyloid. So there is a factor. There is a factor in the serum of these patients. That doesn't happen when you don't have A beta. So this serum from these patients are amyloidogenic. Very interesting study that Andrew did was virus. I told you when the virus reactivates, it causes stroke, or scientific term, vasculopathy. And he took, how do you diagnose vasculopathy? When somebody comes with a stroke, it could be many reasons. Sometimes it is related to the virus, sometimes it's not. So the way we diagnose that is, we take cerebrospinal fluid from the patient and then look for viral-related antibodies. Sometimes you can find virus-related antibodies, sometimes you don't. If you find virus-related antibodies, for example, if any one of us who are not ill, if we take our CSF and look for VZV antibody, we won't find it. Whereas somebody who has a stroke, if you find their CSF and if their stroke is caused by this virus, they will have virus-specific antibodies. Okay? So there are two groups of patients: people who had VZV antibodies and people with stroke that did not have antibodies. So he took the cerebrospinal fluid, looked for amyloid, A beta 42, 40, and amylin. As you can see, statistically significant amounts of amyloid was there in VZV containing CSF, and it contained amylin in these patients, but not in people who are negative. So definitely there is a correlation in the neurological disease. The big challenge in all these studies is how do we manipulate the system? Because this virus unfortunately infects only humans. over the years, people tried many species, so there is no animal model. For a long time, we used cadaveric tissues, and there was an argument that it is a dead tissue. It is not live. It is not like kid having a chickenpox, somebody having chickenpox. So it turns out for a couple of decades, several groups tried guinea pigs, mouse, rats, even immunocompromised mouse. In all these cases, the virus you can inoculate, it'll just have some antibody response, that's about it. No chickenpox, no disease. The virus does not, really sometimes doesn't become latent, and you can definitely not study shingles. So it was very frustrating. And this is something which we didn't know how to handle. And my mentor, Don Gilden, who's a clinician, he got really frustrated because we tried many animals, nothing happened. Today, if he did this, he'd be in jail. He did this in the 80s. He packed a suitcase with virus samples. He said, I'm going to the primate center. I'm going to make sure that the primates will definitely get the disease. The answer was no. He went there, he inoculated, nothing happened. They were completely normal, as if you didn't do anything. They didn't get chickenpox, nothing. So it was so frustrating. However, at that time, we found something called monkey chickenpox. It's called Simian Varicella virus. It's an agent for monkey chickenpox, just like it has fever for 1 or 2 weeks like a kid, and then it occurs in epidemics and it's preceded by an incubation period, just like human disease, but it's a different virus. So that's when we said, okay, this is the only model, and the monkeys suffered. As you can see here, this is just like human chickenpox. This is an African green monkey with chickenpox, very similar to human disease. In both cases, the virus becomes latent. This is a human shingles, and this is an ophthalmic shingles in monkeys. When they get, some of the species of monkeys, when they get shingles, it is very bad to see, horrible to see. It's as if their whole body was on fire. So to prevent other animals from getting that, we have to isolate them. Unfortunately, we may even have to euthanize them so that we can save another 20, 30 monkeys. It spreads like wildfire. So by studying this, we not only help the humans, but also the primates. So overall, both cause chickenpox, both become latent in neurons, both spontaneously reactivate, and they can cross-react. The genomes are very similar. You can use one antibody to cross with another. And when we sequenced, we found that the size, this was sequenced by Davison in Scotland. This was sequenced by our group, very similar in size. So to make long story short, we showed over the years that we can reactivate, produce shingles experimentally by irradiation. Because people who have bone marrow transplant or even cancer patients, they get treated with irradiation or tacrolimus, which is immunosuppressant, or steroids, or a combination of three. So we used those combination over the years and showed that in multiple species, cynomolgus, African green, Rhesus macaques, we can experimentally reactivate to produce shingles. Now, one of the things that is very important in this virus is what's called a CD4 T cell immunity. CD4 T cells are one kind of T cell which protects us. Now, in this case, if you deplete, we showed that if you deplete the T cells, immediately the disease is pronounced. So we know CD4 T cell immunity is important. When you deplete them, it reactivates just spontaneously. So what do we do? How do we study Alzheimer's disease using this model? So first step, we went back to the culture. We took Simian Varicella virus, put it on culture, did exactly what was done with the human virus. And yes, we saw this is the viral protein in the same area, the beta-plated structure, amyloid, and beta amyloid. These are A-beta proteins, and this is the co-localization of all. So just like human VZV, this SVV also induces amyloid in culture. Then, is it amyloidogenic? This is a very interesting experiment Andrew did. This is infection. This was done in New Orleans. Pre-infection, day 4, day 9, day 8. And this is when they get chickenpox. So he took serum and he spiked it with A beta 42 peptide and looked for amyloid. As you can see in the spiked sample, at the same time when the virus replicates, its amyloid is formed. So as I said, this is an amyloidogenic environment. There is some factor in the serum, infected virus serum, that pushes the amyloid formation. And also, this is like a typical chickenpox rash and a shingles rash. And as you can see, this is the chickenpox period, this is latency period, this is reactivation here. So as you can see here, this is virus DNA by PCR, and this is amylin. At the same time, when the virus DNA is there, amylin goes up, it comes down, and then slowly, just before shingles, amylin comes up again. So amylin is also increased in the serum, and it also is an amyloidogenic environment. This experiment can never, ever be done in humans. No mother is going to allow you to get blood samples sequentially. Here, we are able to get blood samples, serum samples, and really look at what's happening. Another interesting thing which I'm not presenting today because of lack of time, we analyzed in this point the sugar, glucose levels. It was all over the place. These primates during chickenpox are diabetic, which we never know in kids. We don't know the kids become diabetic during chickenpox, but we found out that when we look for glucose level, it is all over the place. It increases fasting glucose levels. So tissues. This is pancreas because amylin is made in the pancreas and it goes. So we looked at pancreas. This animal was very sick. It's a primate center in Austin, Texas. They called me and said this one animal came down with severe disease, and they isolated the animal, and then they had 40 different animals. How the virus went into the colony, we mapped the whole thing, where the source of the virus could have been. And then we found out that it was 2 animals that really transferred the virus, and this one animal was affected. The pancreas, this is all, the brown is all virus, and this is pancreatic islets, insulin positive cells. It was virus was present all over the place, inside and outside the islets. Then adjacent, and also we extracted DNA and showed that there are 35,000 copies of virus DNA in the section. Then we analyzed for beta amyloid and Thio-S, found that it was positive, and another amyloid P protein was also positive. So there is tons of amyloid along with virus in the pancreas. Then we looked at other organs. Now it turns out that there is a concept called vascular dementia. Vascular dementia is definitely associated with VZV. VZV infection, there is a strong association with vascular dementia, and 90% of Alzheimer's patients have vascular dementia. Therefore, we looked at arteries. This is a carotid artery. As you can see, this is the adventitia. This had 28 copies of virus DNA. This red color is virus and the green color is amyloid. Both are localizing right outside the artery. Then we also looked at the gut, the intestines. As you can see, this is jejunum, and you can see viral antigen, viral DNA, and amyloid in these animals. And also, not every artery was positive. When you do these experiments, people worry because they say, hey, you see it everywhere, every artery may be positive. Then you can see that there is one artery that is positive, and the same one, this big artery is negative. Now, does the viral antigen need to be there all the time? One would say, oh, you need to have the virus to do the effect. But towards the end, I'm going to tell you that the virus need not be there. And you can wait till the end why I'm saying that too. So, Alzheimer's disease is pathology of the brain. Okay? But, yes?
in the previous slide, when you were showing us the slides, you mentioned jejunum has, you've taken this jejunum in Varicella stage. That means the other two are in the shingle stage.
Oh, yes. We not only, we take this particular animal. This particular animal had varicella because it was part of a big cohort and it was did not have never seen the virus, so it was infected and it became really pronounced. In these two animals, we had established experimentally, it went through chickenpox, it became latent, it was immunosuppressed, then it had zoster. So this is in fact several weeks after zoster. This was an acute infection. Yes.
I'm so sorry.
No, please.
Is there any correlation between becoming latent, then becoming immunosuppressed, and then getting shingles to it being arteries?
Yes. See, that's a very good question. The thing is, during primary infection, the virus is carried by T cells everywhere. In fact, we showed years ago that when the virus gets into the orofacial root, it goes to the bronchoalveolar macrophages. I'll come to you in a minute. Then the virus goes to the lymph node, T cells pick it up, it goes everywhere, including the arteries. On the other hand, in reactivation, it starts from the ganglia. It makes its way along the axons to, when you have an artery, the nerve endings are here, it gets deposited and it irritates. Yes?
So you were talking about CD4 cells. Why is it called CD4?
It's complement determinant T cells. The T cells, in the immunology field, there are two areas. One is B cells and the T cells. B cells make antibodies. T cells are they engulf areas where cells are infected. CD4 T cells called helper T cells. CD8 T cells are called killer T cells. So CD4 T cells produce certain molecules to help the CD8 T cells to engulf. So that CD4 is just that a marker that they find on that surface. CD4. That's why it's called CD4. Complement determinant 4, not 4. So it's a brain disease, but olfactory system is the port of entry to the brain. It turns out that this is the closest, the olfactory bulb and olfactory epithelium are the closest to the brain. And it turns out in Alzheimer's disease, even before cognitive impairment comes, that one of the early signals is lack of smell. One of the early symptoms of Alzheimer's disease is smell problems. So we wanted to know what would happen with viral infection. Just to give you a little bit of education, just like I needed it when I first time I read this. So this is the nose where all the smell, dust, everything goes in. This is called the epithelium. This is called the olfactory bulb. As you can see, this is the trigeminal ganglia. They innervate both. There are nerve endings here, and it is connected to the hippocampus. This organ is the closest to the brain. Okay, so we have, and again here the enlarged. This is the epithelium. This is the olfactory bulb. Okay, so we have a big cohort of Alzheimer's disease patient bank, brain, olfactory system, liver, every organ in Colombia, South America, and well characterized controls. So we were able to have access to the olfactory bulb olfactory tract. So what Andrew did is did ultra sequencing and found that particularly in the Alzheimer's olfactory tract, the viral markers were increased. There was a lot more viral markers than controls. So that indicated that there is definitely the virus has something to do with it. So we wanted to look at in the primates. So what we did in our lab is looked at olfactory bulb in aged monkeys. These are 17, 18-year-old animals. You can see the chickenpox, you can see the virus and amyloid. And this is co-localization. Sometimes they are in the same cell, sometimes there is one, sometimes there is the other. And this is shingles. As you can see, there is less virus, less amyloid, and co-localization. But there is amyloid both during chickenpox and shingles in the olfactory bulb. Now, usually in amyloidogenesis, it's not all bad. Every one of us, to some extent, we would have, as we grow older, we would have some amyloid, and that will be constantly be cleared. That there is a function for amyloid in our system. These are immune defense mechanisms. How are they cleared? They are cleared by these cells called microglia. Microglia are outside the CNS, they are called macrophage. In the CNS, they are called microglia. Microglia engulf those amyloid and destroy them. That is their function. But if you activate them so much, they can be very inflammatory. So we wanted to know how I was going to see microglia. As you can see here, I couldn't stain in the same section because the reagents were not common. So as you can see here, there are lots of microglia. Above one is the marker, and this is amyloid. As you can see, the microglia engulfing the amyloid. And the same thing here. This is microglia, this is amyloid, microglia engulfing the amyloid. So there is microglia activation happening in the olfactory bulb. We are moving towards the hippocampus in these animals to look at what's going on. So what have I told you today? The first part, VZV infection and amyloid burden, as well as vascular disease. AD is potentially accelerating the disease. VZV is accelerating the disease. Both VZV and SVV infection induces amyloid. Then virus infection induces amylin and an amyloidogenic environment. Then I told you that in multiple organs, you can see during chickenpox and zoster, you can see amyloidogenesis. So this provides the best model to be able to study virus-induced Alzheimer's disease. So I'm going to quickly go in the second part about whether the virus is needed. So when something happens, when we didn't see the virus, we were concerned. People said, oh, there is no virus there. You're seeing amyloid. How is the relationship? So this was stuff very recently done by Andrew. What Andrew did was before he started this, he looked at patient samples. He took plasma. I don't know whether I have the, yeah. What Andrew did was let me go back for a minute. What happens is cells, for a long time, we know that any cell, it makes these small extracellular vesicles. These are small bags with proteins, RNA, DNA. It just communicates, the cells communicate the rest. We can have cells in urine, in saliva, in blood, everywhere. Everybody knew for decades. But these viruses hijack that machinery. These are called exosomes or extracellular vesicles. They contain this, they communicate. So what this virus does is, this is a very interesting experiment Andrew did with zoster patients who have had shingles, even months after zoster. He took plasma, blood from them. He isolated these extracellular vesicles, exosomes. And he did the same thing with normal controls, people who did not have zoster. Then he took these exosomes and put them on platelets. Platelets are very important for clogging or stroke. When they aggregate is when you get a stroke, platelet aggregation. So as you can see here, when he added the exosomes from zoster patients, the aggregation was higher. Whereas baseline is here. A non-Zoster patient, there's some aggregation. It really moves up. And even after 3 months after shingles, still it aggregates. So these platelets can be a reason for stroke. Not only that, he also showed that this is interleukin 6 or IL-6, which is really inflammatory. That's the inflammatory marker. As you can see in these cells, the inflammation is very high. So the exosomes from Zoster patients is not only pro-thrombotic or can cause stroke, it is also inflammatory. Now the thing is, what is inside these exosomes? So what he did was, he took neuronal cells, sensory neurons, that's where the virus is. This is uninfected. This is infected. As you can see, these are the virus particles in a neuron culture. He took the whole infected cell culture, he put everything on a target cell, and you can see the virus replicating. However, when he isolated exosomes, in other words, the small vesicles on the supernatant, he isolated them, he dumped them on identical cells. He left them for 7 days, no virus. In other words, these exosomes are not infectious. They don't have the virus. They are just bags that the virus releases. So it's a non-infectious exosomes that are pushed by the virus. And the question is, what is inside these? So Andrew did a complete analysis, mass spec analysis, mass spectrophotometry. There's a lot of proteins which are more higher. It's called a heat map. There's a lot more proteins in virus-related exosomes which are absent in normal, non-virus related exosomes everywhere. But in virus-infected exosomes, there are some things that are more. And there are some things that are more in the normal, non-infected stuff. However, the most exciting thing we had was the virus codes for 70 proteins. The virus is a large molecule. It's constantly. Out of that, he found just one protein. It is 62. That alone is packaged. We were just curious why all this, it had so many proteins. The virus allows only that protein to be packaged. This protein is very important for the virus to replicate. We all know that virus activates a lot of genes. So this was very exciting. So the virus code packages only one. Now, what is the virus doing? What does the virus want? When the virus goes there, it wants to infect, it wants to replicate, that's what COVID did. It wants to multiply. Now, as soon as the virus comes, what is the cell going to do? It's going to stop it. Our immune system is going to stop it. So what the virus does is want to break the immune system. First wants to shut the immune system so that the virus can make its way. And that's precisely what it does. As you can see here, he did, I want to spare you with all the details of this, the ultra sequencing and enrichment score analysis. All the immune cells, the immune markers, IFIT, IFIT M1, M3, all of them are downregulated. These are all immune markers. One of the important things is interferon beta. Interferon beta is a very important antiviral response. Now, what he noticed is when you stimulate it with these reagents, interferon goes up. That's how you stimulate. But when he stimulates and adds the extracellular vesicles, exosomes, you see it brings down the interferon response. In other words, the extracellular vesicles has completely shuts the interferon response so that the virus can come and replicate. Now, as you can see here, we wanted to ask the question, is that 62 alone enough? Or there are so many things inside those exosomes. Is 62 alone enough? So what, in my past life, in the 80s, I had cloned the ID62 by itself to express that protein alone for some other purpose. So Andrew took that and showed that 62 alone is expressed in what I cloned. Then when he added that, you see that IFIT1 is reduced Alpha 2 is reduced, but these are all factors. But as you can see here, the vector alone, it comes down. In other words, that protein alone is enough to shut down your host response. Now, okay, these are all in culture cells. What about in real life? What you see in a flask need not be same as what you see in a human body. So he did a very nice experiment where he first took these vesicles, put it in the nose of mice. Okay? He prepped the mice with the exosomes. In other words, when it goes in, it should shut down the immune system. He came down with herpes simplex 1, which normally infects mice and even kills them. Once he puts first the exosome and then came with the virus to ask the question, is the exosome in a real living being shutting down the immune system, this virus replicates? Oh, that was the question. And as I said, this you have seen before. Here, this is olfactory epithelium. This is the virus replicating. Here, it is just vehicle. As you can see, it is replicating, and this green color is microglia. In other words, it is responding well. It is trying to stop it. Everything is normal. Now, here, he used mock uninfected exosomes. So, not much. But when he put the virus exosome, the green is almost gone. In other words, it shuts down the microglia. Microglia is needed for us to clear things. But these exosomes are completely shutting down the microglia, and the virus is continuously replicating. Now, the next thing is the microglia activated? This is only olfactory epithelium. Now, if you look at olfactory bottle, you can see some viral replication here with vehicle, and we didn't even see any virus in the mock. Of course, we don't expect, but when you put in the exosomes and then the microglia go crazy because it is just completely, they are super activated. So the extracellular vesicle control the immune system. That's how the virus hijacks. It hijacks the whole system, sends these vesicles, and then comes in and starts replicating. And this is yes.
These green emissions that you showed, are these the emissions from these species?
Yes, what it is, yes, very good question. These are microglia sitting there, antibodies bind there, the antibodies has a fluorescent tag.
Which you are putting?
Yes,
Okay.
that's to detect the IBA1 marker or microglia. That's a way to detect. This is a secondary emission.
So that's the reason why it's emitting. It's not that it has an innate color.
No, it has not. We are trying to, like green is microglia, red is virus, and blue is DAPI is, these are all specific antibodies, tags we send there. So, and the virus, this is a quantitation, same thing, but we are quantitating. Anybody else? Yeah.
Just a question. So we can think of the exosomal release as being some sort of cellular deception, where the exosome is sort of mimicking the viral structure itself and hence deceiving.
Yes. Very good point. But then it is happening in every cell. Doesn't matter. Virus doesn't need to be there.
Okay.
It's a cell wants to communicate.
Okay.
There are two cells that want to communicate. Normal, no infection, nothing. It just sends some cargo to a distant location to deliver something.
Okay.
The virus, on the other hand, is a packaged entity. It has DNA, it has protein, it has an axe to grind. It wants to go there, it wants to replicate. The exosomes have nothing to do with replication. They are just going there depositing them.
So is this mechanism something that's built into the viral DNA so that it is better able to replicate? Because in doing this, it overwhelms the immune system because it's resulting in the production of so many other exosomes, which then the immune system then mistakes for being actual viral molecules that it has to.
It can, but the immune system is very specific for viral antigen.
Okay, so it's
So it has to recognize a specific epitope.
okay. So the microglial activity is not entirely analogous with how the immune system is functioning.
Yeah, microglial activity is part of the immune system.
Okay.
That microglia is activated to be able to clear all the garbage.
Okay.
Yes.
Just one question, I'm a bit confused. Do the exosomes contain the packaged protein 62 and that is causing the haywire response of the microglia? That's the only protein
That's the only viral
from
protein.
the
Entire 70 proteins, which we never expected.
yes.
Because why should that protein alone? That's a different question altogether. We are doing many more experiments to find why that. How does the virus decide it's going to package only one?
That particular protein, since you said that the exosomes themselves are not infectious in the previous slide, so that particular protein in isolation is not infectious, but it just triggers the
Triggers, yeah.
the
In fact,
microbial response.
very good. IE62 is called promiscuous transactivator. It will activate everything. So the virus holds the E62 for a reason, and it turns out that other viruses, herpes simplex, 1, 2, they all have an equivalent of 62. It is called infected cell protein 4. So we are looking at that also. This cannot become unique to one virus. Other viruses must also be doing that.
Do we see equivalence beyond viruses as well? So like, do bacteria also have?
Excellent. Yes. Answer is yes. We are thinking that this is not unique to this virus. Very bacterial pathogens or any other infectious agents could be using this machinery also. So, to end the talk, what I've told you today is IE62 or immediate early 62 is packaged into EVs, suppresses the antiviral response, and it increases the susceptibility to other infections. Like when we put that, herpes simplex was able to come and really produce a big infection. Particularly relevant for central nervous system invasion and cell type specificity. Some exosomes are specifically in one organ it can do. These are really fascinating things. Now, one thing that's very important is right now in the vaccine technology, we are always making vaccines to the entire virus. And like COVID, we made the mRNA vaccine, we made the ribosome RBD, determinant domain, receptor binding domain, and we are really interested in the whole virus. But we can make vaccines only 62. We can even prevent the virus from even getting somewhere, because if we stop 62, the virus cannot be as infectious. It can infect, but it may not be as infectious because the immune system will block it. So those are one of the things that we are looking at. How the current vaccine is against virus, viral protein. The shingles vaccine is against the glycoprotein of the virus, which is working very well. So what we are thinking is if we can make antibody response against 62, we may be able to stop the virus from even getting it. The immune system will destroy it. So with that, the most important slide, actual people who did the work, the two amazing assistants I have, Brittany Faya, Malia Stios, these are young women who are exceptional. They do all the immunofluorescence, all the assays for me. The primate center is headed by Dr. Vicky Traynor. These are all the team members who work with all the primates. They take care. I mean, this is a veterinarian. They have taken utmost care to make sure that their health outside the infection is all maintained well. And this is our team. Maria Nagel is our project leader. Andrew, I told you many times, is a faculty member. And Christy is a neuroscientist who did a lot of the exosome work. And Selena is our research assistant. And Dr. Diego Restrepo is like the world's authority on olfactory system. We were so lucky to have him. He guides us with anything to do with the olfactory system. And Dr. Kimberly Bruce is an expert on microglia. And Dr. Seth Frieds helps us with all the bioinformatics, any kind of sequencing, any kind of detailed analysis. He helps us. He is in the University of Virginia, And I really thank you all for paying attention.
Thank you so much, Professor Mahalingam, for the wonderful and insightful talks. The floor is open for questions. Kalyan, please.
Thank you. That's very well said. One thing I was wondering that, and especially with regard to the chicken monkeypox, the Simian version of the virus, there was something about there is no primate model of Alzheimer's disease, although the primates can hit the amyloid, but it doesn't cause the extensive neuronal loss and Alzheimer's.
Yes, very good point. It's a little bit of controversial point. There are some specific monkey species who show amyloid deposition and cognitive impairment. There are some species that publications are slowly coming out. Not all, but the thing is, we don't know when Alzheimer's disease is a progressive disease, right? So a lot of times we compare to human age and monkey age, we have to wait till they get to the equivalent of 70, 80 years old to be able to see. So we are doing an experiment where one of the key thing is in Alzheimer's disease, the glucose dysregulation in the brain is a big, the hypometabolism. And what we have done a preliminary experiment where we did a PET CT scan of these primates before infection and after infection, and the temporal cortex, we are able to see complete hypometabolism. The glucose dysregulation is completely off. So we are just scratching the surface. All these experiments are very, very expensive and time consuming. Each animal is $10,000 and $10 a day to maintain, math. So you can figure, it is so expensive, we are not able to conduct them as freely. Yes, ma'am?
Thank you. It was a wonderful talk. I have a couple of questions.
Sure.
First, in why the virus prefers the ganglia, and the second one is that this protein 62. About the protein 62, you talk about that dodging the immune system. So there are many bacteria deploy multiple ways to dodge the immune system. Have you done any sequence alignment study? Have you found any?
Yes and no. What it is this 62. Let me answer the first question. Why is ganglia targeted? Answer is we don't know. But alpha herpes viruses, the alpha, beta, gamma. The alpha herpes viruses, they always go to ganglia. That's one of the criteria in which we did. Now, what we think is that there are certain receptors in the ganglia. Ganglia, it's a scavenger. Everything broad, folded up. There is a concept called epigenetic regulation, which you would have heard the term epigenetics. The DNA structure is folded by histones and then modified and put away. So that epigenetic regulation we think is important in ganglia for the virus to be put away in this. However, the virus enters via T cell transport. T cells are the ones that are taking the virus, getting into the ganglia. But once it gets into the ganglia, the virus is completely folded up and put away till it has to reactivate. So we are also working on when is the virus unfolded and reactivated in ganglia. But to your question, it is just that the nature of these viruses, we think there are some receptors in the neuron. It can go to many other tissues, but the latency is only in ganglia. Yes, the monkey's liver get affected, lung get affected, but there's no latency. So it happens. And I noticed, in fact, years ago, I showed that the virus becomes latent in adrenals. Now, in adrenal cortex, the medulla are neuronal cells. So it becomes latent. So when I showed in the primates that the adrenals, the virus become latent, immediately my colleague Randy Korsh looked at human adrenals and he was able to see virus. So it has something to do with the neurons. And the second question, IE62. Yes, there are some immune response genes. It has the IE62 protein has DNA binding domain, activation domain, So there are some common things, but we have not explored it. The first time we are showing that it is suppressing the immune system. And yours is a very good question that we can compare to other bacterial pathogens or other viruses which also evade the immune system. We want to look at what sequences are common to these. Yes. Which
Yes, I know, I'm going to ask the same question. You said that the latency is only in the ganglia. Have you stopped looking after the them in the ganglia and they could also be elsewhere? Or are we absolutely sure that it is only in the ganglia?
Yeah, very good question. What he's asking is, are you sure that it is only in the ganglia? Have you looked at all the other organs? The answer is yes. Years ago, when I worked in cadaveric ganglia, my control was lung and liver. My control, ironically, was brain. So I took, the first time when I published a paper about, nobody knew where the virus was. So I was very fortunate when PCR, now PCR is a household term. In the 90s, we started doing PCR. I took about 30 human cadaveric ganglia and corresponding brain and baby, unfortunate death of a baby before chickenpox, their ganglia, and I analyzed all lung, liver, only in the ganglia we were able to see. However, if there is a disease, if there is a VZV-related illness, you can see it in other places. Because the virus has gotten out of the ganglia, it is infecting other tissues. But latency, only in ganglia.
The second question regarding that, back again to the fluorescence thing.
Yes.
You also had some tags which were blue emitting.
Yes.
So if they were blue emitting, you'd have to excite them with UV.
Yes.
Are you sure that you're not causing any photochemical damage during that UV radiation?
Yes, we would probably. So you have a marker. Now these are all very good questions from chemists. So what happens is I have a piece of tissue. I slice it into I slice it into 5 micron or 10 micron sections. So in a 3-dimension, I put it on a glass slide and I bake it so that I get the 3-dimension to 2-dimension. And then my microglia is here, my virus is here, or just the nucleus is somewhere. When I look at the slide, I don't see anything. All I see is a tissue. Then what I do is I add antibody A, which binds only the microglia. And this is, let's say, made in rabbit. This antibody was made in rabbit. Then for virus, I add antibody B that was made in mouse. And then DAPI is a chemical reaction. It's a blue color that it binds. It's a chemical. It binds where the nucleus is. Then even then I don't see the color. Then I come with the second antibody, which is anti-rabbit. It is a goat anti-rabbit. But this has a fluorescent, this has a green fluorescent tag. When you shine UV on it, it will be green in color. Now, for the mouse, I have horse anti-mouse because I cannot use the same species. I use horse anti-mouse, which is red color. Now, once you shine UV on it, the absorption, again, you can tell me the physics of it. So the absorption tells me which color is what. Are you bleaching it 100%? If we keep on viewing it many times, we will bleach it. So we go through the microscope and shine it very short time to take a picture. We can't bleach it if you shine it for too long. So that is, this whole process is called immunofluorescence or immunohistochemistry. I do the same thing without fluorescence. What I do is here, instead of fluorescence, I have an enzyme, either horseradish peroxidase or alkaline phosphatase. There I have a chemical reagent which when the phosphate is cleaved, it becomes green in color to the eye. So I also do immunohistochemistry or immunofluorescence, whichever one works. And everything depends on these antibodies, the affinity of the antibodies. And then one important thing you need to remember, I'm using monkey tissues. Most of the reagents I get is human. Everything is anti-human. So we have to, my two assistants, they have to go through so many antibodies that will cross-react. That's a huge problem in our work because we cannot get cross-reacting antibodies because commercially most of the available thing is humans. Yes, sir.
Just wanted to ask that if it's known biotechnically that how the virus starts the amyloid, as in, so is it the amyloid precursor protein, is it tested? Because there is also a theory that the amyloid beta is involved in the probably the exosome formation of the synaptic vesicle formation in a neuron, and the amyloids are formed outside the neuron.
Yes. Yes.
So
Yes.
it's the APP is cleaved and the amyloids are aggregating outside. So I was just wondering if it's known that how the virus really increases that amyloid.
Yes. Excellent point. At this point, we don't know, but I have a theory. That is, I was telling him this afternoon, in diabetic neuropathy, what happens is, one of the big things is amyloid formation is a huge problem. People who are diabetic can get Alzheimer's more than non-diabetic people. So in diabetic neuropathy, there is this amylin, which is aggregating to amyloid. Okay? Now, but normally in non-diabetic people, there is degradation. Those amyloid is all degraded. But what people who work in diabetes field tell me is that the insulin degrading enzyme is the one that is very important for degrading those amyloid. Okay, so there are two things. One, either you can accumulate the amyloid or you can degrade the amyloid. There's a homeostasis going on. Now, when in Alzheimer's patients, either you're making more and more of amyloid or you're not degrading it enough, right? So what I think is, this is a bias theory, there's no proof, hypothesis. IDE, insulin degrading enzyme, we know that the virus is a receptor. We know that the virus can bind to IDE. So when IDE is doing its job, insulin degrading enzyme is just, in diabetes, it just has to degrade the amyloid. The virus goes and scavenges it away. So it's not degraded properly. So there is accumulation. This is only one of the mechanisms. There can be so many other mechanisms. We are also looking at amylin gene. We looked at the promoter. How does 62 activate the amylin gene? That's a totally different study we are doing. We are trying to fire on all cylinders to find out. Yes.
You mentioned that the property of making these one single protein exosomes might not be exclusive to this virus. So what are there any other viruses that have been studied to show this? Are there any other herpes viruses?
Excellent. Now HIV. This is all very recent. This is only in the last 6 months. I've been constantly combing the literature. As soon as the paper comes, I try to look at, study this. HIV has NEF protein that is packaged, but that's a different mechanism. NEF has a different mechanism. But I completely agree that when a single protein is used like this, it need not be only for this virus. So we also know that herpes simplex virus, type 1 or type 2, type 1 causes cold blisters, type 2 is genital herpes virus, sexually transmitted. Both of them are very similar. And it turns out that its corresponding protein to 62, it's called ICP4, probably may be packaged. The difficulty is the exosome size and HSV size are identical. So it's very hard to study. When you isolate exosomes, you're isolating virus. That is where the catch is. Yes.
So you say that the difference in mechanism, do you think the difference in mechanism of packaging between HIV and something like PCV might come from the fact that this is a DNA virus and HIV is a retroviral?
Very well can do. But the thing is, remember, it is hijacking the cellular machinery. There may be minor differences, but the cell is constantly doing this exosome thing. But the virus is saying, hey, I'm going to put 62 here. It's already existing. The exosome is already existing. But one thing I want you to remember, exosome is not just one single thing. Exosome goes all the way from like 100 microns to 1,000 microns. That's why we call it extracellular vesicles. So I don't want you to take home the message that, oh, exosomes are this small entity. No, there's the whole spectrum. There's 100 microns, there's 1,000 microns. We don't know what is in the 1,000 microns. There may be something else going on. So this is just the tip of the iceberg we are opening up. Yes?
This might be a silly question, but how do you determine if a protein is packaged in an exosome? Like experiment?
Very good question. Not a stupid question at all. Very important. How do we know that a single protein is packaged into this exosome? I really thought a DNA will be there, or an RNA. Yeah, a lot of RNA, microRNA, they're all there. The way we do is, more than one way. One way is you take the exosome, make lots of exosomes, purify them, break them open with soap, and then run a gel, do what's called a Western analysis. That protein will run on a gel and you identify. That's one way. But the amount of, which we are trying right now, but the amount of protein is so small, so it's not able to detect it. The way we did was we took the entire exosomes, we broke it open and did mass spectrophotometry. What mass spectrophotometry does is, any molecule which is in there, it cuts it into pieces. And then when there are protein, it just tells you the sequence of the protein. Then you go back to bioinformatics to find out how the homology, what is that protein is homologous to, and then you link them together and say, hey, the entire 62 is there inside that exosome. That's what we do. In this experiment, we did mass spectrophotometry because that's a very good question. How do we know the entire 62 is there? How do we know that only a small portion the virus is packaging? So we are definitely wanting to look at whether the whole protein is in there. And even more exciting thing, this 62 protein is very well known, and there is a phosphate group. It's a serine 128 or something. If that is phosphorylated, it doesn't get out of the nucleus. That is well known. So the question is, in the exosome, is it phosphorylated? So we are adding phosphatase inhibitors, something that will not remove the phosphate, and then do mass spec to see if it is. Those are all going on right now to see what is the status of this protein.
How big is the protein in terms of?
Oh, this is $120. Huge. It's a huge protein. It's not small. And for whatever reason, it has been a challenge to run a Western to find out Western blot to detect the protein. Yes.
Professor,
I love it when you ask questions.
Professor, do we know what function this protein plays when it's outside of it? It's found in any other.
Beautiful. This protein is called immediate early 62. Now, I'm going to try green this time. So this is the cell. Can everybody see? So the virus hits it. It goes in, it infects, and then the virus replicates, okay? To make new viruses, the virus has a long genome. Yeah, there is a repeat sequence and all that. I don't want to bore you with that. It has 70 genes. And the 70 genes are transcribed in a certain order. There is a set of genes that are immediate early, they are called. As soon as the virus goes in, they are turned on. There is a gene here, there's a gene here, there's a gene, 62 is here. And those genes are called immediate earlies. They are made first. The immediate earlies activate the earlies to make the earlies. And the early genes activate the making of the late genes. The late genes are the ones which are needed to assemble the virus. So there's a cascade. So it turns out that immediate early 62 is one of the very early proteins. When the virus enters the cell, one of the very important proteins is 62 because it activates all the other genes to be made. So in this cascade, you need 62. Yes?
So this protein that enters the cell primarily is what is released out from the exosome of the cell.
Yes.
Also, I had one note. I might use it, but when you were showing the images of the DAPI and the amyloid being detected, you showed that there's a Varicella component and then there's a Zoster component. In the Varicella component, was there more amyloid detected than the Zoster?
Yes, we did see, but it can be purely a sampling.
Okay.
I didn't do like 10 animals and at different time point I didn't do. So it could be sampling, but it could very well be. But Varicella always has more animals. Zoster, you cannot see much. One thing is, Varicella is stored in the body. It spreads everywhere. Whereas Zoster comes only locally. Sometimes Zoster can be only here, and Zoster can be without a rash. So it's very localized. So that's maybe the reason you may not see that much.
Which is why the latency matters, because the latency is only observed in the neurons.
Yes, neurons. Yes, that's where it starts its journey.
Right, let's thank Professor Mahalingam once again.
Thank you.